Good afternoon, everybody. My name is Simran. I'm a Research and Innovation Manager with Genome BC, and I will be moderating this session. So we are in session 7A, Genomics Tools, and our first talk is Funda Ogut on implementing genomic selection in Norway spruce, and she's from Natural Resources Canada.
Hello. Thank you. Today talks, again, it's the implementation of genomic selection in Norway spruce. And then I am based in Atlantic Forestry Center. I'm with Canadian Wood Fiber Center. But we have a collaboration with J.D. Irving, and then this is totally their breeding populations. I will be talking about the genomic selection efforts in their breeding populations in Eastern Canada on Norway spruce. So I will be giving a bit background and then, again, some few cases in terms of genomic selection efforts and then what is next. Norway spruce is a major commercial tree species used in reforestation programs in Eastern Canada. It's not native. It was introduced from Europe over a century ago, and then it was first tested in Ontario, Quebec, and then maritime provinces. It's quite, it has a super growth rate, and then it can survive in drier conditions, and then it has a similar wood qualities with white spruce. The main tree improvement efforts mostly on growth rates with acceptable wood qualities, but it is susceptible to white pine weevil. It's especially it attacks on the young seedlings, and then it kills or damages the terminal leaf, and then it causes growth reduction, stem deformation, and then lower commercial value of the saw timber. So with genomic selection, as we all know, it uses phenotype and genotype data, and then it can reduce the breeding length for any tree species if you have enough good training population. But the goal here is to screen a large number of genotypes for fast growth and weevil-resistant varieties without planting or testing across different environments, which is costly, basically, and then take years and then efforts. So the first genomic selection effort started with the Fast Track project. It was a Genome Canada big project in Eastern Canada. And then a large Norway spruce SNP catalog was developed. And then from that SNP catalog, a Illumina Infinium SNP chip was built. And then that was used for this analysis or the looking at the genomic selection data analysis. So the first data I will be talking about, it's a clonal material. 141 clonal material was propagated by somatic embryogenesis from 19 full SNP families and then 24 parents. And then they were phenotyped at age 13 for growth rates, and then also for the white pine weevil, they were scored with their own protocol. Like if it's no weevil effect, it was scored as 0, and then if there is a current effect, it was 1 passed and then cumulative. So I will be giving some details about this protocol for phenotyping or scoring white pine weevil effect and then how it affects your data analysis. And then, of course, just want to say something. There was a duplicate based on the genotype data, and then it was removed. And then in total, across 4 sites, we have over 4,000 RAMETs. So again, it's a randomized complete block design from 10 blocks. It's a good replicating and then well blocking, basically. 3 sites based in New Brunswick, and then there is 1 site in Nova Scotia. So this is, again, a proof of concept study, looked at the A-blob and G-blob comparison for the predictive ability for this data set. So we have all the trait across site heritabilities, and then we have the genetic correlations across site again. Seems like there is a high genotype by environment interaction, and then for especially the interest for the white pine weevil, low heritability is basically not much genetic variation for white pine weevil. So this might be the reason. The reason might be for the low heritability could be protocol for use for the scoring white pine weevil. The other thing might be the intensity and the history of weevil attacks change site to site. So that's why we don't see much genetic variation in terms of for the white pine weevil. And then again, this is a proof of concept study, and then we looked at the predictive ability of the genomic selection for this clonal material. And then we tried different scenarios, like with the clonal, we assigned 10% of the clones randomly for the validation population, and then for the family stratified, we pay attention to the families, and then we did the we assigned 10% of the clones within family for the cross validation. And as you see here, genomic selection is kind of giving you better or slightly higher predictive abilities, regardless of the whatever the sampling scheme you are using. So it's promising at this for this data set. Then additional material was genotyped in 2021 to start selecting breeding material based on the genotypes only. We're using the same SNP chip as a beyond proof of concept study, basically. And then to continue the implementation on genomic selection, like for Norway's spruce improvement program. Again, there were 576 trees genotyped. Again, no phenotype for this data set. They are coming from 83 FUSIF families and then 8 OP families. And then in total, there are 53 parents, cross 1 to 8 times per cross. Again, this is the validation population, and then only 21 parents are common with the training population coming from the clonal material, which is not ideal. That's why we will see what is happening here. So lots of health SIPs and then unrelated families. With the genomic selection, what you want, lots of relatedness and then connections. So as you see, even though I did some like predictive ability for the as proof of concept for the white pine weevil, I'm not showing because it's not real, basically because of the genetic variance. So I'm showing you here the volume-based accuracy and then its breeding values. The predicted breeding values for these newly genotyped trees are based on the genetic makeup of the clonal material, so 140 trees. And then they are coming from only 24 parents, and then 21 of them are kind of related with this new genotype parents, so which is not ideal. And then as you see, the estimated breeding values or predictions are quite, and then associated with high standard errors. That's why you are seeing low accuracies in here. If you look at these ones, these are the 140 like clonal material, and then this part, especially the cumulative here, those are the ones they have no connections at all with the previous training population. So which is expected. This is a kind of showcase. You shouldn't do genomic selection if you have this kind of material. But regardless, with these results, field testing is required depending on what the industry or the end user needs. So again, this is called beyond proof of concept study, but we need to do field testing to see how we are doing in terms of our predictions. So what is next? Again, in Atlantic, tree improvement. There's an Atlantic Tree Improvement Council. Probably François will give you the detail about that. But it's a standalone tree improvement council across maritime provinces. And then they joined Conifer SNP Consortium to develop and then use available, cost-effective genomic resources for their breeding programs or improvement programs. There is a already developed Norway spruce 50K SNP array by Harry Wu's group from Sweden. And then they allow us to use, since they are joined this group, they can do the genotyping in a cost-effective way, much cheaper. And then the idea here, develop a new genomic selection training population for Norway spruce full-sip breeding population. For this breeding material, it's a 2007 Norway spruce test series. They were phenotyped at age 13. That's the data that I have. As you see, we have no genotype data yet, and then I will be talking just classical quantitative genetic analysis. It's a randomized complete block design from 4 sites, 10 blocks, but this time they put 2-3 plot design. And then in total, there are 84 full-sip families from 45 parents. But as you see, this data is also not balanced. This morning, Brian Cullis was using infill. So not every family is tested across all 4 sites. There is one family tested in only one site, and then there are 6 families or 7 families in only one site in here. And then the only common number of families is 52 across 84 families. So especially you will see the results. Looks like there is a G by E, genotype by environment interaction going for the growth traits. So that means there's a crossover effect, site to site, and then not every parents or progeny is performing the same way. So, and for white pine weevil, as you see, the heritability in here is 0.23. We see something, but they changed their protocol for phenotyping. It's a categorical data still, but there are 9 categories this time. So it's not cumulative like past and present. Weevil attacks is combined, but they looked back, looking at the walls on the trees, and then they quantify and then scored these trees for weevil attack. So thinking and then taking into account this genotype by environment interaction in here for the growth traits, I come up with a sampling scheme across all sites. As you see in here, the breeding values for the parents, but these are the rank correlations. So with white pine weevil and then volume, it's -56. So it seems like if you select for one trait, you will be able to recover the other one. So the goal is selecting the fast-growing and then weevil-resistant lines. So in total, we have 76 families, and then to come up with, in terms of the statistical point of view, we have 8 progenies per site. So it's going to be well replicated and then representing every single site. I dropped a few or 8 families. They present only once or twice in some sites, and then they don't have enough number of progenies per site. And then in total, we are targeting 1,000 or 2,000 trees we'll be genotyping. We also added the parents for this genotyping to be able to reconstruct the pedigree based on the genotype data. With this, again, it's a 50K SNP chip. It's possible to implement. The main goal is implementing the genomic selection, but there might be other chances to implement look at genomic approaches, basically. And then, of course, again, based on the quantitative genetics pedigree-based, we notice we have a G by E, so that's why we need to look at every single site and then quantify. So we might be able to create some H blob, G blob only, and then look at the quantify how they are changing if we have this G by E perspective for the predictions. So we did our sampling, and then waiting for the genotype data, but I have to say, this is my last slide. DNA extraction is the first thing. And then without DNA extraction, your genotyping efforts will be, it's impossible, basically. And then we are already stuck with the DNA extraction because J.D. Irving doesn't have in-source, like lab, to be able to conduct this thing. We sent test trials to a company in the States, but they didn't give us enough quality and quantity for the DNA extraction. And then our first plate, 384 samples, failed, which is a disappointing thing because it's a loss of effort, money, and then time. So now we are working with University of Florida, and then they will be doing our DNA extraction. Then we will be sending our genotype DNA data for genotyping. Hopefully, we will get it this year. That's pretty much it. I just want to thank you, everyone, and then J.D. Irving for their providing their data, and then all the other contributors. Thank you.
We have time for a few questions.
Thank you, Funda, for the excellent presentation. Just for your families, you suppose that are unrelated. Did you check for example, kinship relationship to see, and if it is possible to include it in the model to avoid false positive as we do?
We don't have their parental genotypes. It's first thing. And then when we look at the, like I try to create the pedigree, reconstruct the pedigree based on the genotype data, but there wasn't enough, it wasn't good enough to assign those parents or progeny to the families, basically. But again, that's my assumption, and then I think the reason why we get this low accuracies because they are unrelated, totally disconnected. So that's why we don't see anything.
Thanks, Funda. It was a good presentation. I'm just thinking maybe beyond the actual breeding and start thinking about how do the orchards pass on the seed to the nurseries and the seedlings going back to the land. If we do genomic breeding, but the customers at the end of the line plants at best maybe open pollinated seed, bulk seed out of orchards and so on. It sort of begs the question, why breed like this? If you understand what I'm saying, because you've shown if there's genetics coming in that hasn't been part of your training population, whatever, it sort of dilutes your accuracy. So J.D. Irving has got the capacity to deploy varieties or clones, as they call them. If you really want to capture the value of this, that's the way that you have to deploy. And I don't know if that's ever going to be agreed to. I'm just throwing a rock in the bush, thinking like that.
Honestly, for the beyond of proof of concept, the additional material, I have no contribution how they come up with that material, and then how they decided to come up or use those families, and then because the numbers are also different from families to families. We don't have any phenotype data, so I can't say anything about what would be if they change their scheme or material. So I have no idea. I can only talk whatever I analyzed, and then obviously, based on this analysis, and then for genomic selection, there are some assumptions, and then it's not just forestries, but in plant and animal breeding they have been using. And then lots of successful stories. And then they say, you have to have relatedness. And then if you have this connection between your training population and then validation population, there is no way it's going to work. And then I'm not the one who is going to create the magic, obviously. I'm the last person, basically. But it is what it is. So it's a kind of take-home message, I believe, for them. That's why we come up with this for the new breeding population. what we should do, we can't ignore genotype by environment interaction because you have to populate your training population on the way. With genomic selection, you are not cutting field testing at all. It's just speed up your selection. You have to keep field testing, but maybe not on everything. You can exclude some genotypes, extreme genotypes, at the seed or seedling stage. That's the whole point.
Great, thank you. I think we're going to move on to our next speaker, which is Patrick Lenz, also from Natural Resources Canada. And he will be presenting on optimizing genomic selection for application, the importance of accurate estimation of genetic merit and disentangling of additive from non-additive effects.
Yeah. Hello, everybody. Just thinking about my talk, and first of all, it's always very nice to be at the CFDA and to have the possibility to see a lot of people again for many years and interval, and thinking about what I would present is that I don't feel really old, but I was like, oh, is it already the first time that I will talk about genomic selection? And so just to put us back a little bit is that about 10 years ago, we saw the first genomic selection papers coming out in forest genetics. So Masaru Zen's thesis, for example, then some other studies out in the East, and of course, obviously also Yusri Kasselby's labs and his students who were contributing to the field, thinking about imputation methods for G by S data, and obviously also the G by E questions and so on. With us publishing on Norway spruce and black spruce at one point in time, and then also the colleagues in Sweden working on Norway spruce, obviously, and Nicholas Ukranitz publishing about lodgepole pine. Okay, we try. And but most of those studies were, there was also Bob Thomas's group doing a lot of papers very recently, very good things. But I think it's only recently that we see more examples of papers looking more on the integration of how can I integrate genomic selection in the operational, how can I deploy the gain out into the field, what mating designs do I need, what would work best, and of course also the work that has been done on radiata pine. So just really a quick shot of a few examples in that field. Operationally, what has been done from an Eastern Canadian perspective, you have listened maybe to Funda's talk just a couple of minutes ago. Yes, we have been doing some decisions, for example, on seed orchard management using genomic estimated breeding values. We have been also doing or integrating in the Quebec white spruce program genomic selection into basically the entire program. And these guys are planning doing this forward selection, genomic selection mode with clonal reproduction. And I'm really looking forward what we'll get out of those results in a few years from now. They are currently genotyping several thousands of additional trees beyond the proof of concepts and the genetic studies that we did. So GS, everything is clear. I just put up this slide again. It's a two-step process, obviously. And in the fourth step, for the proof of concept, we were looking at the first step here, most often. And we would basically sample, get a representative sample of our breeding trial, get the phenotypes that we are interested in, get the genomic profiles, the market profiles, and model them. And we can evaluate that model, but beyond proof of concept, we also need to think about the application when we want to predict seedling or embryos based on the genomic profile, and we want to estimate this genetic or breeding value for breeding or propagation purposes. So overall, this is supposed to speed up the selection cycle and augment the genetic gain per unit time. We can reduce to some extent the field testing, but also what is very interesting in view of climate change and getting much more complicated phenotypes into the equation is that we can, especially for those traits, increase our selection intensity if we can only measure so many trees for this and this phenotype. So if we really want to think about application, is that we need genetic relationship. This was Funda's message just a couple of minutes ago. But we also need accurate predictions of our genetic variances. And we are coming up very quickly with the questions, how many samples, how many families? Is it possible to predict untested families, untested crosses? Is this really possible with our markers? So this comes up to the objectives of the studies that we did and I'm presenting to you today. So we really wanted to evaluate and compare the ability of GS and pedigree-based models to petition additive and dominant variances in the right space. We wanted to investigate the effect of sample size on genetic parameter estimates, and we wanted to know or evaluate how does predictive ability change when we try to predict in untested crosses. So, this is a material. We needed a few Genome Canada finance projects to assemble all those data. I'm showing you here a map of southern Quebec and eastern Ontario. This is a little bit where the material, the parental material is coming from. In the end, we are looking about and this data set I'm putting up today is 94 subfamilies, 2,500 trees about, coming from 4 breeding groups. And what we measured, height, growth, and wood quality traits and got the genotypes on an Illumina custom-made array that is and after all the filtering, we were working basically with 4,200, 300 markers from as many gene loci. So GS modeling, I'm not putting up the formulas. I let this job to Brian Callis. We did basically GBlob estimates, which is a linear mixed model. And what we were using is additive and dominance relationship matrices, which structures this random tree genetic effect, basically. And so we are also doing, obviously, cross-validation. I'm going to talk about predictive ability or prediction accuracy, which are both measures to evaluate how well our predicted breeding values or genetics values actually are. So the first set of results, and I'm putting up here, is on heritability. What I'm showing here is 2 traits, height growth, density, wood density. A-blob is a pedigree-based model, G-blob is a marker-based model, and the additive effect is the yellowish color, the bluish is the dominance component. And so first thing what we see is A-blob gives higher heritability. What we also see is that dominance is somewhat lower in wood quality traits, and so far so good. But what we also see is that when we come from an additive-only model and we add the dominance components, that there's a lot of change and variance. So variance seems to switch places. And this component is a bit smaller, or quite smaller, when we're using marker-based models. So this is, I think, a good news for me. What is happening when we are looking at different subset of different families? So what I'm showing here is, again, height and wood density. Narrow sense heritability, broad sense heritability, and the dominance component here in the middle. So different subsets of families from 12 families to 72 basically in this graph here. The red is the pedigree-based, and the bluish, greenish one is the G-blob-based estimates. And especially what we see is at a low number of families, there's a lot of error associated to our estimates, obviously, which is normal. And there's a lot of variation also, especially in the narrow sense heritability, what we don't see further down if we try to optimize or get more families and more connection into our data set. So, just to keep in mind that we need a little bit more families to obtain stable heritability estimators for pedigree-based models, and also that we think that GBlob can potentially handle more partially related families or parents in a population. So what I'm showing here in this is precision of genomic selection models. I'm having, again, here my density and my tree height. Two things that I'm showing. This is a prediction accuracy of breeding values, so the additive part, and the prediction accuracy of genetic values, including the dominance. So this is a darker gray parts. And what we can see here is that, yeah, we have higher accuracy for wood quality traits and compared with growth. But what we also can see is that as soon as we get the dominance component into the models, we can even better predict the breeding values, the additive breeding values, which is good to know. And then we also notice that as soon as we compare the accuracy of genetic values to the initial accuracy of additive-only models, that we are a little bit lower. And this is mostly due to that our dominance estimates are more imprecise than the additive part, the estimates of the additive parts, which is relatively normal. But this comes also with the statement that we need enough crosses to well estimate the dominance component. What I'm putting up here is, you see the two tables here. This is different cross-validation scenarios. So the left one is the prediction into families. If I try to predict in untested families where I only have half the relationship, at that point in time, obviously the accuracy goes down, and accuracy goes down. But what we also see is that when we are putting the dominance into our models, is that the GBlob models, the accuracy of GBlob models is relatively higher compared with ABlob, and which this is especially true for height growth and growth spur properties, which have more dominance as wood quality. And so there's a clear advantage for GBlob if I want to predict untested families, which I can't do actually so well when I'm only relying on the additive part and mid-parent value, for example. Yes, GS enables us to predict at least a part of dominance. So let's have a quick glimpse on the effect of the sample size on accuracy. Is that this is predictive ability, predictive accuracy. I'm not going too much into detail with that, but what we see is that, first of all, the curves are going up with more families tested. This is a good sign. Also, errors become smaller. And so what we really see at a low number of families are these horrible errors around the estimates, and the accuracy from one sample to another is just changing a lot. So let's get as much as family as we can is a little bit the message there. So yeah. So many crosses, simply many combinations of parents to simply get the exact estimate. It's also for the dominance components in here. So the last slide of results that I'm putting up here is the comparison between pedigree and genomic-based estimates of breeding values. So here is the ABlob estimates, the GBlob estimates. This is breeding values, this is genetic values. And these colored dots here are actually the trees that you would retain selecting based on GBlob, but that you would call or not retain based on ABlob. So although the correlation is not too bad, we are missing out on quite an impressive number of samples which would not be culled or selected in one or the other scenario. And so for breeding values, the overlap of retained or culled trees is 64%, which is not too bad. But other than that, if we go to genetic values, it's only half. And so we really need to think about which is the best opportunity. I don't obviously have the answer to that, but this is just need to be kept in mind. So overall conclusion, how am I doing for time? Okay, that's good. So what we have seen is the superior ability of GBlock to separate additive and dominance effects. And so, but you really want to precisely estimate additive. Also, if you're thinking, okay, I'm only deploying an open pollinated seed orchard, but you really want to make sure that your additive value is correctly estimated as correctly as possible. And we have also said that we need sufficiently large data sets so that we can really pin down the additive and especially the dominance variance. And so the better accuracy of GBlock for prediction in untested families is also open as a possibility for mating allocations. This is something that we can't really do with pedigree-based selection because we are relying on mid-parent value, basically. So this is some of a really nice application, in my opinion. And so I just want to say a special thanks to my colleague Simon Nadeau, who was pushing many of those analyses I was just putting up here. The papers come with it. Sorry if you have already read it. I didn't expect this coming out so quickly. But so just look back on that if you have any questions. 1 minute, if I have 1 minute. I got many questions. What are you doing next? What are you doing next? So the next, hopefully I'm going to talk to you about this in 2 years. And the next meeting is, we have a Fast Track 2 project, obviously. So our aim is to bring genomic selection into black and red spruce breeding programs out east. You know, black spruce for you is, I don't know, probably a weed, not really growing very quickly out here west, but in eastern Canada it's quite an important species. And same with red spruce, and especially in the maritime provinces. So what we are trying to do here is to work together with the provinces of Nova Scotia, New Brunswick, GDI here, and also natural resources in Quebec, of the province of Quebec. Jean Bousquet from Laval University and myself leading this project, and I hope that I have next year or in 2 years more results on that. So it always takes a village to do genomics. There's a lot of funding involved, a lot of tech, professional help. I didn't even try to put all those people here on the list. Thanks for your attention.
All right, we have time for a couple questions.
So thanks very much for the presentation. You showed a lot of analyses on the changes in your parameter estimates, increasing the number of families, but did you look particularly at increases in the precision of the dominance variance with increasing number of crosses per parent and increasing number of samples per family?
The short answer is yes. The longer answer is I can't remember exactly. But the thing is that in this data set at the time when they were doing the breeding groups, they wouldn't even think of genomics. And so the common practice at the time was, okay, let's cross each parent 3, 4, 5, 6 times, but not only one time. So, and as soon as you're doing the subsampling, especially also the tree number level that I didn't show today, then you're losing this connection in your cross-validation, and then your precision and your error scale go up. And this is a little bit what we see also when we are going down to a too low number of families, which is no real breeder would do, but we see surprisingly a lot of scientific or research project working with a low number of families. I've done this myself in wood science, or if you work with traits that you can't really measure very well, obviously. But it should be good practice to try to just boost this number of family to get those combinations and don't lose them.
Thank you, Patrick. Nice talk, and a pleasure to meet you here in Vernon. Do you hear me?
Continue.
Yeah. Just for genomic selection or other models based on additive effects, we are trying to explain 95% of the genetic variation in our population, for example. And adding more complex, for example, dominance effects to the model, just trying to explain maybe, as you said, there is no good amelioration in the accuracy. Is it necessary, for example, to add more complex variables in the model? Just to explain, that is my first question. And the second, for non-dominance effects, there is epistasis interaction also. Did you look at that also to include it in the model?
Give me a data set that I can use to analyze that. Epistasis is really hard to grasp, even in model trees, in model species like in poplar and all these cloned and in fruit fly, and so it's really difficult to pin epistasis down. And I would really think a lot of trees. There's sometimes studies out that do this, try it, but I'm a little bit, how do we estimate it? We can all use these squared matrices and so on, relationship matrices, but I'm a little bit, how to say, anxious for overestimating, trying to constrain too much, or trying to constrain too much variance with all the parameters to estimate. So I'm not sure how useful this is on our data sets in forestry. If we would have really well-crossed material, cloned many times, maybe we could come back to that question, but it would need really tremendous data sets, in my opinion. And so this comes back a little bit to your first question, is that obviously you always look at AIC, log likelihood, which is the best model, and is it worth to do more and sophisticated stuff or add dominance? And yes, indeed, sometimes the difference wasn't big, especially for wood quality traits. Dominance is slower, so there wasn't a real need to get the dominance in. But as soon as you get growth in GBlob models, it was helpful.
Great, thank you. And so we'll move on to our next talk. We have Dayoung Lee from the Seoul National University, and she's going to be presenting on comparative transcriptome analysis reveals putative genes involved in cone development in Korean red pine.
Is that my presentation? Yeah, hello, my name is Dayoung Lee. I'm a PhD student at Seoul National University, South Korea. Actually, it's my first oral presentation in English, so I'm a little bit nervous. So, yeah, the topic of my presentation is comparative transcriptome. Okay.
I want people to really hear you and talk together.
Okay. Sorry. My topic is comparative transcriptome analysis reveals putative genes involved in cone development of Korean red pine, Pinus densiflora. Let me start with the introduction. For seed plants, reproductive development is essential for survival and breeding. Perennial trees have a long vegetative cross period before reproductive cross begins. But unlike angiosperms, which takes about 1 or 2 years to flower, conifer trees have a very long reproductive period. So it takes about 3 years to make complete cones. Because reliable reference genomes and mutations in gymnosperms are lacking, genetic studies on the flowering process in conifer species are insufficient. Conifer trees have a long breeding period, so it takes decades to develop new varieties by traditional breeding method. As such, it is difficult for slowly growing trees to respond to the rapidly changing demands. If improved varieties are developed, for example, by inducing early cone development, progeny tests will be advanced and the overall yield of cones can be increased. Pinus densiflora is a conifer species which is well adapted to dry and barren environment. It occupies the largest area as a single tree species in South Korea. So in this study, we compared the expression of transcripts in various tissues, including the female and male cones, during the cone development of Pinus densiflora. Tissues were sampled from other trees in the experiment forest of our university. As you can see from the table, 3 stages of reproductive tissues were sampled during the cone development period. And the female cones are sampled a week later than male cones. In addition, vegetative tissues, including apex, needles, roots, and stem, were sampled once from the same trees. And all the tissue type was represented by 3 biological replicates. RNA sequencing, in short RNA-seq, is defined as NGS technology to determine the presence and quantity of RNA in biological samples. Using RNA-seq, the structure of protein coding genes can be analyzed, and new genes with specific functions can be discovered. We can perform gene expression quantification and comparison by treatment or organ using RNA-seq data. De novo is a Latin term that means anew or from the beginning. So de novo assembly means a bioinformatic method for linking and assembling sequencing data, so-called read, to create whole transcriptomes. We can use this analysis when there is no published reference genome. This is the pipeline of RNA-seq data analysis I used in this study. Gene co-expression network analysis is an undirected graph composed of nodes and edges. And here, in this figure, each node corresponds to a gene, and a pair of nodes is connected with an edge if there is a co-expression relationship between them. And half genes are defined as highly connected genes within a module. So we use this analysis to select the target gene clusters modules and identify half genes associated with the cone development. Quantitative real-time PCR is PCR that can quantify DNA or mRNA in real time. So the initial amount of DNA or RNA can be determined by measuring threshold cycle, CT, which is the point where the threshold and the amplification curve intersect. QRT PCR was used to verify expression levels of a subset of differentially expressed genes, TEGs. Hierarchical cluster analysis was conducted to investigate the differential expression among reproductive and vegetative tissues. This analysis showed tissue-specific clusters, indicating the statistical significance of 3 biological replicates. However, female cones were not completely clustered at each stage of development, and this is probably because the growing rate of female cones differed among the biological replicates. In addition, late cones were separated from other stages of male cones, and this is because last sample was collected in a state where all the pollen was blown. So, yeah. And to identify disease, we compared the transcriptomes of 10 different tissues. And total 2,507 genes were identified as disease in the all pairwise comparisons. And the comparison between root and male cones showed the most disease. And the comparison between apex and female cones showed the least difference in gene expression. We found that 4,212 genes showed sex-specific expression in only female and male cones. Functional GO enrichment analyses were conducted using the disease found in female and male cones at 2 different developmental stages. In female early versus female late, lignin by synthetic process was significantly enriched in early female cones, and plant type cell wall loosening was enriched in late female cones. On the other hand, in male early versus male mid, lipid catabolic process, syncytium formation, lignin by synthetic process, and pectin catabolic process were significantly enriched in early male cones. But however, there was no significant GO term in mid-male cones when compared with the late male cones. Also, functional enrichment analyses were conducted using the disease between early female and male cones. In early female cones, GO terms related to plant hormones and flowering was predominantly activated, and in early male cones, lipid metabolism and cell wall related genes are predominant when compared with female cone early. And these are the results of gene co-expression analysis. Red module was positive for female cones, and blue module was positive for male cones. We visualized the selected modules and identified hub genes associated with reproductive development. A gene encoding agamous-like 6 and a gene encoding MIB domain protein 80 were identified as the hub genes, respectively. To validate that the expression levels of transcriptomes were accurately measured using RNA-seq, qRT-PCR was performed in this study. Total 12 diseases, up-regulated and down-regulated both in the female and male cones compared to stem, were randomly selected and amplified. With a correlation coefficient of 0.74, we could assure that the results of this study, which were analyzed and interpreted by RNA-seq data, were reliable. Let me summarize the main points in this study. Conifer species have sex-specific reproductive structures that are ovule-bearing female cones and pollen-bearing male cones. In this study, each developmental stage of the female and male cones of Pinus densiflora showed different gene expression characteristics. Based on the results in this study, the biological process of each cone development can be summarized. In the early female cones, lignin biosynthesis was predominant. It is reported that the thickening and lignification of cells are necessary for opening of cone scales and providing resistance to decay. On the other hand, in late female cones, cell wall loosening is activated. Cell wall loosening is required for expansion or major cell wall disruption. And it is reported that cell wall loosening of the mature pollen grains occurs at the surface of the stigma during pollen-stigma interaction. Through these processes, the female cones eventually become seed cones. In the case of male cones, some different processes were predominant in the early stage of male cones compared to the later stage. Syncytium refers to a multinucleated cell structure. And in a major structural process in early pollen development is the syncytium formation of microspore mother cells, MMCs, followed by separation of each MMC to create microspores. And in fact, the walls of mature pollen grains are composed of intins containing pectin and cellulose components, and exins composed of sporopollenin, a lipid-based polymer. Finally, we found half genes of female and male cones. Agamous-like 6 was the half gene of female cones in Pinus densiflora. Agamous is a badge box family gene required for normal flower development in Arabidopsis and confers the identity of the floral organs. And this gene is known to be expressed in the ovule layer adjacent to the growing female camelophyte. So therefore, it can be suggested that Agamous-like 6 plays a central role in regulating a transcriptional network during the female cone development. In male cone development, MIB domain protein 80 was the half gene. And this transcription factor is necessary for pollen development and the regulation of programmed cell death in tapetum in Arabidopsis species. Tapetum is the inner cell layer that's around developing microspores within the anther and is necessary for supplying nutrient and structural components. As the pollen matures, the tapetum undergoes PCD, providing many molecules required for pollen development. And it is reported that some downstream genes of MIB80 are important for male cone development in gymnosperm. So this gene is assumed to play a major role regulating a transcriptional network during the male cone development in Pinus densiflora. So that's all I prepared for you today. So thank you for listening.
All right, are there any questions? All right, I guess you're free to go.
Oh, thank you so much.
All right, and so we'll move on to our next talk. And we have Akriti Bhattarai from the University of Connecticut, and she will be speaking on identification and classification of potential disease-resistant genes across the North American, European, and Asian white pine species.
That's the laser pointer. That's forward, okay. Hello everyone, good afternoon. My name is Akriti and I am a master's student at the University of Connecticut. Thank you for this opportunity to talk to you about my project identifying, classifying disease resistance genes across white pine species. So to start, I'm just gonna give a brief overview of the white pines and white pine blister rust, the genomic and transcriptomic resources that I use here in my project, and then the overall NLR identification process that I used, and then the results of that. So the white pines comprise the subsection strobus of the genus Pinus. Subnotable species here in North America include eastern white pine, sugar pine, and white bark pine. White bark pine is listed as endangered here, I think, in Canada, and recently was listed as threatened in the United States. So that's a really ecologically important species. And so obviously the reason why we care about this is not just for commercial purposes, but also for the overall health of ecosystems. And white pine blister rust is caused by the fungal pathogen Carnartium rubicola. If people who went on the trip yesterday might have heard a little bit about this. It was introduced to North America and thought to introduce to North America in the early 1900s. And it causes these sort of like canker blisters on the stem. And it over time will kill trees, often kills young trees very quickly. And since it's been around for a while and there have been a lot of efforts to understand white pine blister rust as well as disease resistance mechanisms in certain species. And so research programs over the couple of decades have identified major gene resistance in 4 of the species here in North America. And some of the candidate genes for major gene resistance have been identified as NLRs. And I know I mentioned that in my title and also a little bit earlier in the overview, but what are NLRs? NLRs are nucleotide binding leucine-rich repeat receptor genes, and so these are the largest class of disease resistance genes within plants, and these are generally pathogen specific, so like gene to gene. They're considered to be like a gene to gene analogs, and so they have a very characteristic structure consisting of an N-terminal domain, a nucleotide binding domain, and a leucine-rich repeat. You can kind of see where it gets the family gets its name from. And the N-terminal domain can be one of 3 types. I'll talk a little bit more about these later, but these are also used to classify the NLRs into subfamilies within the overall family. So in terms of the transcriptomic and genomic resources that I used, so for the white pines, there are genomes, the genome assemblies are only available for 2 species, one of which just recently became available and will be publicly available very soon. But in order to do a sort of like a greater comparison across the white pine species, I used RNA sequencing samples that were generated from this phylogenomic analysis of pine species, of global pine species. And I took those RNA sequencing samples from this paper and from a couple other papers. I reassembled those transcriptomes so that I could get a representation of the gene space. But like I said, we do have genome sequences available for Pinus labicalis, which is the white bark pine, and then Pinus lambertiana, which is sugar pine. This phylogeny over here also shows a little bit of the, I guess, the basis of knowledge that we have about resistance and susceptibility to white pine blister rust in different species. There isn't much validated anything on the Asian pines, but they are thought to have co-evolved with the pathogen, and so the assumption is that they are resistant as well. All right, so for the white pine genomes that I was able to use, like I said, we have the sugar pine genome and the white bark pine genome, Pinus lambertiana, Pinus labicalis. The sugar pine genome has been available for a little while, version 1.5. The estimated genome size is estimated to be about like 31 gigabase pairs. However, the assembled genome is significantly smaller than that. And however, we believe that it is due to filtering or collapsing of reads during scaffolding. However, it does have quite good N50, so it's a very contiguous genome. The Busco score completeness here is talking about single copy orthologs, and so the higher the Busco completeness percentage, the more likely that you have a more complete genome. And the white bark pine genome just recently finished annotating that, and will hopefully be up on Tree Genes soon. And so this was also used for identifying NLRs, and I'll talk a little bit about the difference between the transcriptomes and the genomes as well. And before I move on, just talking about the types of resistance to white pine blister rust. So Pinus lambertiana, sugar pine, does have major gene resistance, CR1 allele. But white bark pine, Pinus albicollis, does not. However, it has been shown to have significant quantitative disease resistance, and there is a wide range across, I guess, the range of white bark pine families as well. And so the identification of NLRs is not just about candidates for the major gene resistance, but also these are going to be genes involved in quantitative resistance as well. And so my process for identifying and classifying these genes started off, well, after I assembled the transcriptomes, I used both protein and coding sequences. So I started off with the protein sequences, and I used two programs, InterProScan and RGAugury. InterProScan is used for scanning protein domains, if anyone has used something like that before. RGAugury is more of a specific pipeline for the identification of NLRs that was developed in about 2016. The reason why I used InterProScan and RGAugury is that I could not identify one of the N-terminal domains with RGAugury, and InterProScan doesn't have as well of coil-coil domain identification. And then with the coding sequences as well, so they're the same sequences as the protein sequences, but just before it's been, instead of being peptides, just the nucleotide sequence. And I use this program called NLR Annotator to scan these coding sequences for NLR-associated motifs. And then these motifs were then combined together to create proxies for those protein domains. And then I combined the results of these three different methods to create a more complete annotation of the NLR space, and also to help resolve some ambiguous annotations. Maybe one program was able to identify one or two of the domains, and another program helped provide the identification of the last domain. And so this was for the transcriptome-based. And so what was different about the, all right, after that, after I've combined them all together, I get my predicted NLRs. But what was different about the, with white bark pine and sugar pine with the genome annotation, I used the genome annotation instead to provide the protein sequences rather than using the transcriptome. And then I also used the whole genome sequence, and I used NLR Annotator as it was intended to be, as a whole genome scanner to identify potential NLRs. And then that was then overlapped with the genome annotation, and results from NLR Annotator that overlapped were then considered part of that genome annotation NLRs. And so after identifying them, I classified them based on their N-terminal domains. They're broken up into the TNL, CNL, and RNL subfamilies. And then also those with all three domains were identified as complete. So here are my results from the transcriptomes only. So I identified a total of 5,071 NLRs across these 22 transcriptomes. And here is just showing everything is colored according to their native region. You can see, I did not mean to skip that. I meant the laser pointer. All right, so you can see that white bark pine is right here, had the lowest number of identified NLRs. And then sugar pine is right here. It is actually quite high performing in terms of the number of NLRs I was able to identify. But one thing to note is that of all of the transcriptomes that I used, sugar pine had the deepest sequencing, which I believe is part of the reason why I was able to identify more NLRs in there. There weren't very clear patterns with tissue type or very clear patterns with resistance that I was able to see using the transcriptomes. And then here I'm comparing just the genome results for white bark pine and sugar pine. So here, using the genome instead of the transcriptome, white bark pine went from last to pretty close to the top. And you can see here, for sugar pine, I greatly increased, like almost double the number of identified NLRs And then also the number of complete NLRs that I was able to identify were greater. However, just in general, overall, I was not able to most of my NLRs were incomplete. They also could not be classified because they were missing their N-terminal domain, things like that. So there's a lot of caveats with using the transcriptomic resources for something like this. There can be everything from problems with the transcriptome assembly to not have just the identification methods as well. And then, so comparing again, my results with using the transcriptome versus using the genome, you can see here when I use the de novo assembled transcriptome for white bark pine versus scanning the genome directly versus using the genome annotation. And here, the reason why the genome scan over-predicted a lot of NLRs is because there's a possibility of including a lot of false positives and a lot of pseudogenes. Whereas with the assembled transcriptomes, it all comes from RNA sequence evidence, so expression-based evidence, and the genome annotations were also mainly based off of RNA-seq evidence. And then to talk a little bit about, specifically in white bark pine, something that was really interesting that I was able to do using the NLR identification method and the genome annotation. So in genome, so say this is a scaffold, and these are the complete NLRs that I was able to identify with NLR annotator. Again, some of these may be false positives. And then overlapping the genome annotation, that was mainly based on RNA sequencing support. And then those that overlapped with the NLR annotator results were considered as NLRs. But we also had an ab initio gene prediction used for annotation. However, we decided not to include that because there were a high number of false positives and a lot of smaller or fragmented gene annotations. However, using the results from NLR annotator and the complete, NLR annotator can identify complete versus partials as well. And so using the complete NLRs from NLR annotator overlapped with the ab initio gene prediction, I was able to pull in more NLR genes, and this also could be used to augment other annotations as well. And right here, sorry. So to summarize, the combination of protein domain and sequence scanning methods was required to identify all of the different NLR types. I was able to identify 5,701 potential NLRs just using the transcriptomes. Even with really deep sequencing of the transcriptomes, the genome sequence and using the genome annotation greatly increased the number and the quality of the NLRs that I was able to predict. And genome scanning with NLR annotator could be used with ab initio gene prediction to recover more likely true positive NLRs into the genome annotation. And with that, I'd just like to thank everyone, thank my advisor, Dr. Joe Wegerson, and everyone who's helped me out in this project, and all of our collaborators on the white bark pine genome assembly and annotation project.
Thank you, that was great. Are there any questions?
Thanks for the great talk. So do you expect that NLRs are expressed sort of constitutively, so like at all life stages, at all points, or only when they're challenged by the pathogen?
There might be very, very low expression, but it's very, they are expressed in response to pathogens, which is also another thing with, using publicly available sequencing data. I don't know exactly what conditions, what those tree samples were exposed to, what those tissue samples were exposed to. So that was also another kind of caveat, is that it wasn't specifically inoculated and then the response to inoculation.
Perfect, thanks.
Great talk. So I also have a similar question. Are there any plans to rerun this analysis with whole stem samples that have been or are currently experiencing infection with C. rubicola?
Not like across all the species. I am, so I do have like hopefully an RNA-seq comparative study between Pinus sibirica, the Siberian pine, and Pinus labicalis, white bark pine. Still working on the RNA extractions for those, and it's been like 3 months and I still can't get the protocol to work. I'm mostly a computational person, but hopefully I could do more of like direct comparison between at least those 2 species they were inoculated and non-inoculated samples. It really is so finicky.
Any other questions? All right, well, thank you. And I want to thank all of the speakers for this afternoon session. And we have an announcement.
Just briefly, those of you who are going on the Friday field tour, could you please meet in the lunchroom just at the start of this break? Thank you very much.